Hi, I am Abby Bunkum. I'm a PhD student in the Computational Cancer Genomics Lab at University College London, and today I will be presenting a novel method called SPICE for the probabilistic reconstruction of copy number evolution in cancer. So tumours develop as a result of a complex evolutionary process, where over time, tumour cells acquire different genomic alterations, such as single nucleotide variants or copy number alterations, resulting in heterogeneous subpopulations of cells with distinct genomic profiles. In order to model this evolutionary process, phylogenetic trees have been developed in order to model the evolutionary relationships between these cells. And the importance of identifying phylogenetic trees in the context of cancer is that it may enable us to identify drivers of tumor progression or genomic alterations involved in the development of drug resistance, as well as to infer metastatic migration patterns to determine how the cells move around the body. One of the signals that can be used to infer phylogenetic trees are copy number alterations, which either increase or decrease the number of copies across the genome. And the way in which we can model copy number evolution is to look at the number of copies across the genome in different tumor clones, as we see here. And we can use this to determine the relationship between these clones and whereabouts these copy number events occur during evolution. So for example, we can see here that there's an event that is found in all tumor clones. And this here is one that could occur truncally in tumor evolution. And so several methods currently exist to reconstruct the evolution of copy number alterations and enumerate a phylogenetic tree. However, copy number alterations are actually incredibly complex events, and they have the possibility to overlap, which can make the inference of phylogenetic trees from copy numbers incredibly difficult. And there can actually be multiple plausible phylogenetic trees possible for a single tumor. However, existing methods currently don't account for this uncertainty and select a single phylogeny by methods such as either parsimony or likelihood. And so in order to address this issue, we've developed a novel method called SPICE, which aims to reconstruct phylogenetic trees and enumerate all of the multiple equally plausible solutions. With our method, we're able to enumerate thousands of solutions and use this to determine the uncertainty within our phylogenetic reconstruction. In particular, we can also look at the probability of copy number alterations occurring at different times during tumor evolution. So on the right here, we can see multiple phylogenetic trees where we have a copy number alteration denoted by the letter X that can occur either in early branches as shown in red or in later branches as shown in purple. And by determining the frequency at which we observe this event within different trees, we can look at the probability of this event occurring either early or late. And we can apply this principle across the entire genome. Specifically, SPICE takes an input copy number clone profiles and uses an integer linear programming model that aims to minimize the number of copy number alterations within the tree. We can enumerate completely unique solutions that are both optimal and suboptimal. The copy number evolution model that we use incorporates overlapping or interval events, which are large events that span large regions of the genome, and this is a principle first introduced in MEDIC. We can also account for whole genome duplication, as well as pre-whole genome duplication events and post-whole genome duplication events within a single phylogenetic branch. We can also account for allele-specific copy number alterations, as well as loss of heterozygosity. So this phenomenon is where once we reach 0 copies of an allele, we know that we can never regain this, and this can help us to infer parent-child relationships within our data. So the first question we wanted to ask is sort of how important is it for us to reconstruct these multiple solutions? Do we find that there are many optimal solutions within the data? And so in order to achieve this, we applied SPICE to 20 simulated data sets and enumerated 1,000 solutions per data set. On the left here, we have a plot that shows us the number of solutions that we observed, where the y-axis is the relative number of events compared to the optimal, such that 0 is the optimal solution. And what we can see is there are actually thousands of optimal solutions possible for even these 20 data sets, highlighting the importance of us accounting for these within our phylogenetic reconstruction. We then wanted to look at the impact that this could have on the accuracy of our phylogenetic reconstruction, initially looking at tree topology accuracy, where the Robinson's fold distance indicates that we've inferred the correct topology, and also the ancestral node copy number accuracy, so how good we are at inferring these internal copy number profiles. And so as we can see here, a couple of important things actually. So firstly, we see that there are many suboptimal solutions that have a correct, both in terms of tree topology and also in terms of ancestral copy numbers. And this highlights to us the importance of not only enumerating optimal solutions, but also these suboptimal solutions that could also be correct. Another important thing we see is that there are solutions that are optimal or very close to optimality that are actually much further away from the ground truth. And this is important for us because if we were to enumerate just one single solution, it could be that we selected one of these solutions, which would be entirely inaccurate. From here, we wanted to look to see how incorporating phylogenetic uncertainty could improve our inference of copy number evolution compared to existing state-of-the-art methods. So when we look at WGD accuracy, tree topology accuracy, as well as our ability to infer ancestral node profiles, we can see that by incorporating this phylogenetic uncertainty, we get a better reconstruction of copy number evolution. We then wanted to look at how we can use SPICE and apply our method to real data, so specifically 51 metastatic samples from 10 prostate cancer patients, first analyzed by Gundam et al. In 2015. And here we have an example of the results on one patient. So this is patient A22, where we apply SPICE to clone copy number profiles that were inferred using the Hatchet tool. And we can see that we obtain hundreds of solutions for different tree topologies. When we look specifically at one topology, we can see that there is a large variability in the copy number profiles at certain genomic regions. As we can see, there's a high number of regions that have completely different copy number profiles for different solutions. And this is a demonstration to us that SPICE is able to capture this uncertainty, and actually that there is such a wide range of uncertainty within even the real data here. And so I'll now demonstrate 4 important applications that are enabled by SPICE's application to this data. Firstly, looking at our improved estimation of copy number alteration frequencies. So as I mentioned earlier, one of the uses of SPICE is that we're able to determine the probability of copy number alterations occurring. Whereas existing methods that enumerate just a single phylogeny could only identify an event as being either absent or present in different patients, as we can see here, where the event is present in only a single patient. When we use SPICE and look at the probability of the events, we can see that there's actually a probability that this event occurred in multiple patients. And this is useful if we want to look at the frequency of events across the cohort and aggregate our patients together, because we can incorporate this information to get a more accurate idea of how frequent this event occurs. We've applied this principle across the entire genome, looking specifically at the presence of copy number losses that occur truncally. And what we can see is that there are many differences when we use a single phylogeny versus when we can incorporate multiple phylogenies, specifically in the case of chromosome 4 here, where if we use a single phylogeny, we don't observe any truncal chromosome losses for this section. Whereas when we incorporate multiple phylogenies, we can see that there is a probability that these events have occurred. And this result is corroborated by previous analysis that also identifies clonal chromosome losses within this chromosome. So now that we've used SPICE to look at improving our estimation of copy number alteration frequency, we can next look at characterizing the presence of unexpected events. And so for this analysis, we've looked at genes that are known to be important for driving prostate cancer progression, specifically tumor suppressor genes, which are frequently lost during cancer progression. And here, when we use a single phylogeny, we can see that we observe many truncal amplifications of tumor suppressor genes. Now, this is a result that we wouldn't expect because the loss of these genes is known to drive cancer. Having them amplified seems to be counterintuitive. However, when we incorporate multiple phylogenies, we can see that for a majority of these genes, the chance of a truncal amplification occurring is actually very low. And this highlights the importance of accounting for this uncertainty to give us an idea of the confidence we have at specific events occurring. Next, we can look to identify parallel copy number alterations. Now, parallel copy number events are those which occur on distinct phylogenetic branches and multiple times throughout tumor evolution. And this is important because if an event is occurring multiple times, it suggests that the gene that is impacted by this event may be important to drive tumor progression. Here we're looking specifically at the parallel amplification of oncogenes. And we can see that using a single phylogeny, there are a few cases where we observe parallel copy number alterations. However, when we incorporate multiple phylogenies, we can see that there's actually a probability that many more of these events occur that we would have missed had we used just a single phylogeny. Finally, I want to show how we can identify high probability metastatic migrations using SPICE. So as I mentioned previously, one of the important applications of SPICE, reconstructing phylogenetic trees, is that we can infer metastatic migration patterns. And this is feasible thanks to a tool called Machina, which takes an input, a phylogenetic tree, and reconstructs the possible migrations between different tumor sites. We can apply this principle to the results of SPICE, where we have multiple phylogenetic trees, and we can run Machina and determine metastatic migration patterns for each one of these solutions. We can then condense all this information and determine the probability of migrations between different tumor sites. So here, where the light-colored arrows denote migrations that have a low probability, whilst the dark-colored arrows are migrations that we're highly confident in. And this is important because we can look at the solutions that are responsible for the migrations we observe. So at the top here, we have a solution which has 399 events, and this is the optimal solution that we obtain. So it has the minimum tree cost. However, when we look at the migration probabilities from this solution, we can see that they're actually very low, and this is unlikely to have occurred. Whereas when we look at one of the trees that is less optimal, it has a couple more events, we can see that the migrations resulting from this are much more likely. And so to us, this tells us that accounting for these multiple solutions is important to give us an idea of high probability metastatic migrations. And in particular, accounting for these suboptimal solutions too may help us, because it could be that the less optimal solutions give us migrations that are more highly probable. And so to conclude, today I've introduced SPICE, which is a novel algorithm to reconstruct copy number evolution and enumerate multiple plausible phylogenies. I've also demonstrated the presence of many different equally optimal solutions and shown that these are really important to help us improve our reconstruction of copy number evolution. I've characterized the phylogenetic uncertainty in order to enable robust identification of both early and late copy number alterations and parallel evolution. And then finally, I've shown how we can use multiple phylogenetic trees in order to infer the probability of metastatic migrations between different sites. So I'd like to thank my entire lab, particularly my PhD supervisor, Simone, as well as all of our collaborators and funders. Thank you so much for listening.
Nice presentation, and we have a time for questions.
Hi, excellent talk. Thank you. I was wondering, how does the SPICE take into consideration variance while modeling the copy number evolution? And if so or not, how does it compare to variant methods?
Yeah, so we don't take into account single nucleotide variants within this model. I think if we were to incorporate variants, I think the models are very different and it would make it significantly more complex, such that we might end up getting it would be harder to reconstruct, and we may have to make more assumptions that would make our inference worse. However, there are obviously methods that do reconstruct variant evolution as well. And I think one of the sort of really exciting uses of this could be, for example, in the case of reconstructing metastatic migrations, where we could reconstruct migrations from a copy number phylogeny and then also an SMV phylogeny. And this may help us, for example, if we see a migration that's supported by both these signals, that gives us a really high confidence that this actually occurred. So I think this would be a really exciting potential use to incorporate both.
That's great. Thank you.
Thanks.
Great talk, super interesting. I was wondering about your whole genome duplication and timing those.
Mm-hmm.
How do you call whole genome duplications in your data set? And do you think that impacts like how you'd guess the order of events if things are like large events all at once, but not a whole genome duplication? Could that change your phylogeny?
Yeah, so we model whole genome duplications as this is just one event that's possible, and obviously we penalize for that within our model. So if you were to infer a whole genome duplication, that would be the one that gave you the minimum events, and then obviously we would enumerate all the other solutions. And so I think by enumerating the different solutions, we'll be able to investigate that question to see whether if it's a whole genome duplication or whether it's large events, and sort of how frequently we see the different possibilities and the impact that this has on the overall phylogeny. So I think that would be really exciting.
So it's like something you're calling within.
Yeah, exactly. Yeah.
Thanks.
Thank you.